Well, good afternoon. Welcome back to Virology. Today we're going to get in the nuts and bolts of the infectious cycle. That is what happens when a virus infects a cell, what goes on, and how we can tell that something is going on in an infected cell. So when a virus gets into a cell, we'll talk about these steps in great detail throughout this course. We call this the infectious cycle. Everything that happens from the start to finish in an infected cell, we call the infected cycle, infectious cycle. Sometimes you'll see it referred to as the reproductive cycle, means the same thing. And we like to divide this into stages. And in fact, that's what I do in this course. I divide it into attachment and entry, translation, genome replication, assembly, release. Of course, in an infected cell, everything happens in a continuum. There are no labeled segments of the infectious cycle. We just do that to make it easier for us to study. Just like we have nomenclature for viruses to make it easier for us to understand them, we divide the infectious cycle into stages. And through the first 12 or so lectures, we're going to look at each of these steps in the infectious cycle and understand how they work in different virus-infected cells. But before we get into all of that, we need to make sure you understand what I mean when I say a particular word. You know, the English language is famous for having ambiguous words, but that doesn't work in science. You need to know what I'm saying. And here are some of those examples. A susceptible cell. What that means in virology terms is the cell has a receptor for the virus. So on the cell surface, there are proteins for the cell's own purposes. Viruses have evolved to be able to bind to those receptors and use them to get into cells. And the presence of a receptor that works for any particular virus makes that cell susceptible. Okay? But it says nothing about the ability of the cell to support the viral replication or reproductive cycle. A resistant cell has no receptor. That's easy. A permissive cell has the capacity to replicate the virus. So that's different from susceptible. Susceptible is the receptor. Permissive is if the genome of the virus, the RNA or the DNA, could get into that cell, would it reproduce and make new virus particles? That's what permissive means, or permissivity. All right, now, many people I know, many virologists, don't even recognize that these words are what they mean. So a lot of virologists and scientists in general are not too much of sticklers with their terms. I am, because this textbook that is associated with this course, when I wrote this many years ago, you have to use the same definition for a word in chapter 1 as in chapter 25. If you change it, it's really bad. So we had to establish a terminology. And so that's what we have. And finally, a susceptible and permissive cell is the only one that can take up a virus particle and replicate it. So I want you to know, when I talk about permissivity, exactly what I mean. It's very easy. It's the ability of the virus to replicate in the cell. Think of it this way. If you took the nucleic acid out of the virus particle and you inserted it into the cell, and we have ways to do that chemically, bypassing receptor at all, that would measure permissivity. And that's what we're talking about. Now, in the very beginning, when viruses were first discovered, remember at the end of the 18th century, tobacco mosaic virus, the early 1900s, we started to discover animal viruses. There were no cultured cells. Today we have cell cultures that allow us to do a lot of things, but back then there were no cell cultures, so people propagated viruses in laboratory animals of all sorts. Mice were very common, but chickens, primates, non-human primates, dogs and cats, whatever worked for a particular virus, and often it wasn't the same host as the virus it was originated from. Of course, if it's a human, you can't grow viruses in humans, so you have a real problem. So people used laboratory animals. And a very common laboratory animal was the embryonated chicken egg, shown here. So this is a chicken. This is a kind of egg you would have today if you like eggs. You crack it open, and there's a lovely yolk in it, and you cook it. And the proteins coagulate, denature as you cook it, and it turns white. Well, this is the same thing, except there's an embryo in it. It's been fertilized, And this is about a 10 to 12-day-old chicken embryo. If you let it incubate, it would eventually hatch. And in the old days, virologists used to use these to grow viruses in. And you can see you can inject viruses into different places. Chorioallantoic membrane, amniotic inoculation. The amniotic sac is the one that surrounds the embryo. Then there's allantoic cavity is the bigger one in blue. You can put viruses in there. And you can see all the different viruses that were grown. Pretty much we don't use this anymore except for influenza virus. We do use this to grow influenza virus vaccines and also influenza virus in the lab. I did my PhD thesis on influenza viruses, I don't know, over 40 years ago in the 70s. And we used to grow the virus in eggs. And we had an incubator, a walk-in incubator that you would take your trays of eggs and put inside to incubate for a couple of days. And if you forgot about them, they would hatch into chickens. And then we would come in the morning and see little chickens running around in the warm room. So this is pretty much old technology, but it really worked well for these viruses back then. You could grow a lot of virus in these eggs. Today, as I said, a lot of the flu vaccines are grown in eggs because it's way easier than growing them in cell culture. In cell culture, you have to grow up hundreds of thousands of liters of cells. You have to keep it sterile. You have to harvest. It's really hard. The egg comes sterile, and it stays that way. The way you grow a virus in them, if it were just me, we used to have a little Dremel tool and drill a hole in the shell. You'd stick a syringe in, inoculate the virus, and then you'd have some hot wax that you had melted. You'd seal it over. And that would keep it pretty sterile. You'd wipe the shell first with ethanol before you inoculate it. Now they have machines that do this automatically. There's an automated machine where you put racks and racks of eggs, it takes them in, it drills holes in them, inoculates them with virus, and then it incubates them. And this is how we get the flu vaccine. So next time you get a flu vaccine, most of them are grown in eggs, not all of them. About one egg goes into your shoulder in that shot. However, we don't do this for most viruses, and most of the time we don't grow viruses in animals any longer. We have gone beyond that. And that's because of this discovery made by this virologist, John Enders, cover of Time magazine. You ever see a virologist on the cover? Probably never saw Time magazine to begin with, right? But have you ever seen a virologist on the cover? There actually have been a few, because he got the Nobel Prize in 1954 for this. He was the first to show that you could grow poliovirus in human cell culture. No one had been able to do that before. 3 people did this together, and they all got the Nobel Prize for that in 1954. This revolutionized virology. Wow, we could grow viruses in cells and culture. We don't have to use animals, which were a real problem. So today we have all sorts of different kinds of cell cultures that we use to grow viruses in. And here are 3 different kinds. We have what we call primary cell cultures. This is where you take a tissue, and it could be human or animal. You chop it up with scissors, you digest it with trypsin to make individual cells, and then you plate them in cell culture dishes. And they will grow for 20 to 30 cell divisions. But because cells don't live forever, because the ends of their chromosomes get shorter and shorter, the telomeres, eventually they will die, and you have to go back and make some more. And this is a primary human foreskin fibroblast, because foreskins are thrown away on a daily basis in hospitals. Right? And so people who work near medical centers can go and get them. You can go in with an ice bucket and get a bunch of foreskins. Now there are probably companies that sell them, but you used to do that. But so for some things, a foreskin or a primary culture is great, but often you don't want to keep making them over and over again because they're not going to be consistent from batch to batch. So we have found out that you can make cell lines, and sometimes these are immortal. They live forever. So here we have a mouse fibroblast cell line called 3T3. And then we have the famous human epithelial cell line, HeLa cells, the first immortal cell line ever made, and many people use these. These are great because they grow forever. You can grow them in your lab and keep splitting them over and over and over, and they will outlive you if you treat them well. The problem is they're weird. They have weird numbers of chromosomes. They have all sorts of mutations. So depending on what you're doing, they may not be perfect. If you want to just grow stocks of virus, probably okay. But if you want to study what the virus is doing in the cell, probably not, because these cells lack many genes, they have too many copies of other genes, and they may not give you the right answer. These are called continuous cell lines or immortal. We also have other kinds of cells which we call diploid cell strains. So these have the right number of chromosomes. A very famous one is called WI38. And these don't last forever, but they do last longer than primary cell lines. So there's something in between, if you will. So here's how you grow cells in our laboratory. We have lots of cells in culture. You have an incubator, which is at 37 degrees, and you put cells in different kinds of plastic vats. You have dishes and bigger dishes, multi-well plates, little flasks and big flasks. And the cells are all growing on the plastic. They adhere to the plastic, and they have nutrient medium on top of them, which has serum in it and other goodies to help them grow. And this incubator is given 5% carbon dioxide to act as a buffer so that as these cells are metabolizing, the pH remains constant. You can tell by the color of the medium. Now, some cells grow in suspension. And this is unusual. Most cells do not, but some of them can be adapted to grow. And here is a flask where we're growing these in suspension in an incubator. And it has a magnetic stir bar in it, and underneath it is a magnetic plate that is spinning it. And you can get really, really high densities of cells in these kinds of cultures. So if you need a lot of cells, this is the way to go, because this is probably a few hundred huge tissue culture flasks worth of cells, much easier to make here. Now, many of you may recognize this book, which was written by Rebecca Skloot some years ago now, The Immortal Life of Henrietta Lacks, which tells the story of HeLa cells, which were taken from a woman in 1950. She had a cervical tumor, and she went to the Johns Hopkins Hospital. They took a little biopsy of her tumor, and they actually kept it, and it grew into HeLa cells. She eventually died, but the cells live on to this day. We use them in the lab. But no one told her family about this, and they found out many years later, and this book is the story of that. And it's really, really nice. It's a really nice story. Okay, let's do a quiz. The infectious cycle. A blank and blank cell is the only cell that can take up a virus particle and replicate it. Fill in the blanks. A, naive and resistant. B, primary and permissive. C, susceptible and permissive. D, susceptible and naive. E, continuous and immortal. Most of you got C, susceptible and permissive. That's right. So those two words, susceptible, having receptors, and permissive, being able to replicate the viral genome. Now, when you have a cell line of some sort that you're using to grow your virus and you infect it, how do you know that your virus is growing in it? Let's talk about that a bit. One of the first things people did when they first made cell lines, John Enders, he took poliovirus, which was grown in a monkey most likely, and he put the virus on his cells in culture, and he saw that they died. That was the most obvious outcome of a virus infection. The cells just die, and that's what's shown here. This is a series of panels of cells infected with poliovirus. This was done in our laboratory many years ago. And here on the top left, you can see a lovely monolayer of cells. So when cells grow in a sheet on a layer of plastic in a dish or a flask, we call it a monolayer. And then we've infected them with poliovirus, and different times after infection, maybe 2, 4, and 8 hours, you can see at 2 hours, you start to see a few cells getting round, and they're detaching, and then the next time point, most of the cells are floating, and then at the end, many of them have actually broken open. And this is called cytopathic effect, or CPE. And it's just a way of saying, I've infected these cells, I'm looking at them under a microscope, and I can see changes. And the changes that happen are all different depending on the cell and the virus. This is one kind of change, rounding up, detachment from the plastic, and dying, the cells dying, but there are many others as well. Another kind of change, cytopathic effect, is when cells fuse. So here we have cells in culture. On the left is a photograph, and you see some single cells, and these have been infected with a virus that causes the cells to fuse together. And you can see in the photograph one large cell with lots of nuclei in it, and that's because a lot of cells have fused together to form that. This is called a syncytium. So syncytium is singular, syncytia is plural, and many viruses can do this. And the way it happens is you have a virus-infected cell, and on the surface of the cell when the virus is replicating in it is synthesized one of the viral proteins that the virus would normally use to get into a cell. And that causes fusion with the virus and the cell during infection. But if it's on two neighboring cells, so the red Y is the viral protein that's catalyzing fusion, it attaches to a receptor on a neighboring cell, those two cells are going to fuse. And on and on and on and so forth till you get this big syncytium. And so that's characteristic of viruses that can encode these fusion proteins. So just a list of some different kinds of cytopathic effects. This is not something I would ever ask you to remember. I'd never ask you on a test or anything to tell me, what kind of virus causes rounding up or detachment. I just want to give you a sense for the large diversity in the amount of cytopathic effects that we see. So here on the left we have the different kinds of alterations, and on the right are the viruses that do them. And these include morphological alterations in the cells, so rounding up and detaching, as I said before, vacuoles in the cytoplasm, syncytium formation, breaking up of chromosomes. You get the idea. And then also inclusion bodies. This is another kind of cytopathic effect where different viruses cause very specific inclusions in the cell, which could be virus particles, either in the nucleus or the cytoplasm, factories where virus particles are being assembled, clumps of ribosomes, and so forth. And some of these are very diagnostic for a particular infection. For example, Negri bodies. This was an inclusion in a rabies virus infected cell noted by an Italian pathologist many years ago named Negri. And he said, wow, this is happening in dogs and people who have rabies. And so it turned out many years later we found out that that was caused by viruses in the cytoplasm. So that's the first thing you can do, is look at an infected cell and see changes and you say, aha, it's infected. But you want to go beyond that, of course. You would like to know how many viruses the infected cell is producing. And that gets us to quantitation of viruses and virus particles. And we can divide this into two broad categories. We can measure infectivity. So I can take a supernatant from an infected cell and ask how many infectious viruses are in it. Or I can actually measure the physical particles of the viruses or parts of them. We'll talk about a couple of examples to illustrate this. In my opinion, the best infectivity assay in all of the world is the Plaque assay. I love the Plaque assay. And this was first developed in the 1930s for people who were studying viruses of bacteria, bacteriophages. And here is an example of that. And you may have done this even in high school because it's a relatively safe assay to do. You mix virus and bacteria and you plate it out on an agar plate. The bacteria grow, that's the cloudy background. And then wherever a virus is killing the bacteria, you have a clear zone. And that zone of killing arose from one virus particle. So you can count the number of plaques and you can get an estimation of the number of viruses in a sample. We call this plaque-forming units per milliliter. So 1930s, it was first developed for bacteriophages. 1952, this gentleman, Renato Dubecco, developed it for animal viruses. This was his paper published on April 29th, 1952. He was at Caltech. Production of plaques in monolayer tissue cultures by single particles of an animal virus. You may wonder, how does he know there were single particles in 1952? Well, I'll show you. He didn't look at them. And he got the Nobel Prize in 1975 for a number of discoveries, including this one. So this is, of course, very important for animal viruses. So the way you do a plaque assay, you have a solution of virus. I give you a tube of virus and I say, tell me how many virus particles are in it. So we start with our virus stock and we make 10-fold serial dilutions. Very simple stuff. You put a little buffer in your tubes and you just transfer 0.1 ml into 0.9 in each case, so you're getting 10-fold serial dilutions. And then you have to sort of have an idea of what your titer is going to be. If not, you use all of your dilutions. But here we kind of knew how high titer this virus would be. So we put 0.1 mLs into 3 different cell culture plates. So we have here plates with a monolayer of some cell, which we know is susceptible and permissive to our virus. We infect, and then we will cover these plates with an agar overlay and incubate them and get plaques. How that happens, I'll show you in a moment, but eventually you can stain the monolayers and you can see the plaques very clearly. There are holes in the monolayer, just like clearing of bacteria by bacteriophages. And we can count the plaques. Here there are too many, and here there are too few on the right to be statistically relevant. But in the middle, we have a good number. We got 17 plaques on the dilution of 10 to the minus 6. But remember, there's another tenfold dilution here. 0.1 ml is another tenfold. So that's 10 to the 7th dilution. So 17 times 10 to the 7th PFU per mil, or 1.7 times 10 to the 8th. And that's the titer in plaque-forming units per mil. So what we're saying is this tube has that many in it that will form this number of plaques. We don't say virus particles because we can't say that, and you'll see why in a moment. Now let me just illustrate plaque formation in a few ways so you understand what's going on here. So on the upper left, we have our cell monolayer, and we've infected this with our dilutions of virus. And in these plates, one virus is going to sit down on a single cell and cause an infection. And that virus will maybe kill that cell, so it's shown missing from the monolayer on the left side. It's going to release new viruses, and those are going to affect neighboring cells, which will then die, and those will in turn infect more neighboring cells. So we're getting a bigger and bigger hole in the monolayer made because the cells are dying. Now remember, there's an agar overlay on top of this monolayer, which is restricting the diffusion of the virus. That's why you get discrete plaques. If you put a liquid overlay on top, the virus would go into the liquid and spread throughout the culture, and the cells would all be dead in a day or two, and you wouldn't be able to count any plaques. So that's the importance of the agar overlay, to restrict diffusion to neighboring cells. Here on the bottom are two close-up micrographs of plaques. On the left is a plaque caused by a herpes virus, and you can see the surrounding monolayer, and then right in the middle, the cells have detached and rounded up. And if you take the overlay off and stain this monolayer, you'll get a clearing right there where all those cells are washed away. On the right is the same virus, but this time we've put a gene in it that makes a blue dye. All right, so instead of needing to look for cell destruction, sometimes viruses don't kill cells very well, but you can visualize a plaque or a focus of infection by incorporating into the virus a gene that'll make some kind of color. In this case, it's a color, it's a substrate that we add and it turns blue. Now here is a movie of a plaque formation, which came out about 8 years ago. Before that, I had to do a lot of talking to describe what happens. It's a time-lapse movie where they infected a monolayer, and then they identified a small focus of infected cells, and then took a frame every so many hours and put them all together. And you can see we're up to, I think this is hours up here, 14, 15 hours of incubation. And you can see it starts as a small focus of dying cells, and it gets bigger and bigger. It's kind of like you drop a pebble into a pond and the ripple goes out. So these are all dying cells. It illustrates the virus release and spreading, and eventually that'll be a plaque when you stain your monolayer. Virus, plaque formation, the movie. Greatest movie ever made, in my opinion. Now in my lab, we do lots of plaque assays. I love them, and my people love them too. And this is an example from one experiment that someone in my lab did years ago. She did 1,600 plates. These are 6-well plates with 6 wells of cells, and we did a plaque assay in each one. And she had all these in the end. She said, "What do I do with them?" I said, "Well, you have to build a wall." So she built one in the lab, but the fire inspector said we couldn't keep it there. So I moved it to my office, and it's all glued together. And I had to leave a spot for the light switch and the thermostat. If you come to office hours, you can see this. And I have a page on my website. People come and they like to get their picture taken in front of it. And I have some quite well-known people, like Jonas Salk's son came and so forth. So I call it the wall of polio. And I think it's really cool. And a lot of people, I will post this on social media, and people go, "What is that?" And then that gives you a teaching moment, so you can start talking about plaque assays. So that's really why it works. All right, our next question is, when doing a plaque assay, what's the purpose of adding a semi-solid agar overlay to the monolayer of infected cells? To stabilize progeny virions, to ensure that the cells remain susceptible and permissive, to act as a pH indicator, to keep cells adherent to the plate during incubation, to restrict viral diffusion after lysis of infected cells. All right, let's see. Most of you got E, to restrict viral diffusion. And that's right. Remember, you put a solid overlay so the virus particles will be restricted to the neighboring infected cells. If you had a liquid overlay, the viruses would spread throughout the culture. Now, a couple of you said to keep cells adherent to the plate. So the cells would adhere anyway. So under liquid, they would adhere because they're an adherent monolayer, so they don't need agar to keep them down. But of course, if you had liquid, the virus particles would come out of the cells, diffuse in the liquid, and spread throughout the culture, and you couldn't count plaques. So that's why we use a semi-solid overlay. And just parenthetically, there are all kinds of agar you can buy out there with different compositions, and viruses are sometimes susceptible to what's in the agar. You know, this is a product of seaweed, and it can be purified to different extents. So often, if you have a virus and it doesn't form plaques, just try different agars, and often you will hit it. It's a matter of hit and miss. So here's another stained plaque assay, 6 plates. We do the dilutions in duplicate, of course. And here you can see they're paired vertically, and you can see nice 10-fold dilutions. This happens to be influenza virus. Now the question, is it really important? How many viruses are needed to form a plaque? Because why do we use PFU per mil instead of virus particles per mil? So we can figure out how many viruses are needed. Remember in Delbecco's paper, he said by a single virus particle. And the way you do that is really simple. You do a dose-response curve. And a dose-response curve means you make dilutions of something and you look at what happens. And you could do it with drugs or you can do it with viruses. Basically on this one, we have an XY plot. The x-axis is relative virus concentration, where we're increasing from left to right. So these are just dilutions plotted in the opposite way. And on the y-axis is the number of plaques. And so what you could do experimentally is simply make a series of dilutions of your virus, plaque them out, count the number of plaques, and then the kind of line that results, well, whether there's a line or a curve, tells you whether you need one or more viruses to form a plaque. So here, the red line goes through the origin. It's a straight line. That is a line which we call one-hit kinetics. You need one virus particle to infect a cell and cause a plaque formation. And that's because for one-hit kinetics, the number of plaques is proportional to the first power of the concentration of the virus. Basically, if you double virus concentration, you double the number of plaques. That's why you have a straight line. So Delbecco did this with his animal virus plaque assay. He got a straight line, and that's why he concluded that you needed just a single virus particle to form a plaque. Now you can see also on this graph we have a curve, the blue one, which is not a straight line, and that is the curve for 2 hits. You need 2 virus particles to form a plaque. All right, and the math is there. The number of plaques is proportional to the square of the concentration. It turns out that some viruses have 2 pieces of genomic information, and they're packaged in separate particles, and you need both of them to infect the cell to start an infection. So that's an example of 2-hit kinetics. A lot of plant viruses are like that. We're not sure why, but the fact is they have multiple genomes, some proteins on one genome, some on another. They're packaged separately, and they have to both be infecting the same cell. But most viruses have 1-hit kinetics, so one infectious virus is sufficient to cause a plaque. Now, the plaque assay was also used and still is used to what we call plaque purify viruses. You take a stock of virus, you do a plaque assay, and then you take a pipette, you plunge it into the agar over the plaque. You're going to get a little plug of agar in your pipette, and then you can expel that plug into some buffer, and that plug of agar will have virus in it. So the virus that gave rise to that plaque, if it were a single virus, you're now clonally purifying that virus. Now, why would you do this? Well, if you've just isolated a new virus from an animal or a human, and you want to make sure it's a pure culture before you start working on it, you could do this. And we usually do it 3 times just to make sure we're not getting overlapping plaques. Could happen. And we make clonal virus stocks that way. Now, sometimes viruses don't form plaques. No matter how hard you work and try different agars and agaroses, you can't get plaque formation. There are other ways that you can quantify infectivity. One is called an endpoint dilution assay. And these are done typically in 96 well plates or something like that is shown here with multiple little wells. You put cells in each well, and then you make dilutions of your virus. Here we're going -2, -3, -4, all the way to -7, and you infect each well with a dilution, and you can do it in multiple samples with multiple replicates. You see here across the top of the well, each of those wells gets the same dilution, so you have many replicates of -2, -3. And then you incubate the cultures and you see which wells show cytopathic effect. Now, you may be asking, well, if the virus causes cytopathic effect, why isn't it forming a plaque? That's a great question, and it happens. It may be that whatever agar you choose, it's inhibitory to the virus forming a plaque, but in liquid, the virus can kill cells. And then you can simply say, what's the dilution of virus where half of the cells or half of the replicates are killed? And so that's shown at the bottom here. Here, -2 dilution. The plus means all of the cells showed cytopathic effect. You could stain these 96 well plates and see very readily if there were cytopathic effects. And now moving down the dilutions, you can see here at -4 we have one well that didn't show any CPE. So that means we're getting to a dilution where there are fewer and fewer viruses, so we're not going to infect every well. And then -5, you can see there's a mix. And then at -6 and 7, you don't get many wells infected. So you can see the effect of diluting out the virus. Now it turns out that at -5, there are half of the wells are infected and half are not infected. And so the 50% endpoint would be 10 to the 5. We would call these tissue culture infectious doses 50%. Most of the time, you're not lucky to get the 50% endpoint on a particular dilution. It usually falls between two dilutions. Then you have to do some math to figure it out. So that's endpoint. That gets around viruses that don't plaque. Now let's talk about this thorny issue, what we call the particle-to-PFU ratio, because up to now I haven't told you this, but not every virus made in an infected cell is actually infectious. Many of them are not infectious. Even though they look like a normal virus particle, they're not infectious. That's the particle-to-PFU ratio, and it's defined by the number of physical particles divided by the number of infectious particles. So the number of physical particles, you'd have to way of counting them, like looking at them in an electron microscope or using some other assay, and then you do a plaque assay. And what we found out years ago is it's not one-to-one often for most viruses. An infectious particle may be rare in a population of viruses. So what this means is that the one-hit growth curve means that a single particle can initiate infection, but not all viruses are successful in doing that. And that's because they're damaged, they have mutations. The infectious cycle is complicated, and if it fails at any one point, it's not going to be the proper particle. Whatever the reason, to this day we really are not sure, makes doing virology complicated, because you never know, depending on the virus you're working with, you never know if the physical particles are actually having the effect that you're looking at. So let me illustrate this with some examples. So here's a table of particle-to-PFU ratios for some animal viruses. And here at the very top, papillomavirus. These are DNA viruses that cause warts of various sorts. And the particle-to-PFU ratio is 10,000. That means for every plaque or every infectious virus in a preparation of this particular papillomavirus, there are almost 10,000 that are not infectious. So these infected cells are churning out not infectious viruses, only 1 in 10,000. Of course, that's enough to infect the cell, but the others are not infectious. And you can see the number varies, 30, 1,000, 50, and so forth. But some viruses are close to 1. This one down here, an alpha virus, 1 to 2 particles per infectious unit. So that's not bad. You can work with that. But you can imagine, let's say you infect a cell culture and you do some microscopy and you're looking at viral proteins in the infected cell. How do you know that those are the infectious ones that are doing whatever it is that you're looking at? It makes things complicated, and you have to figure out how to get around it. That's something you have to think about. Our next question is, in the particle-to-PFU ratio, particle can be best described as one of the proteins which makes up the virion, a virus which may or may not be infectious, a virus which is infectious, a virus which is not infectious, elementary or composite. You see this word virion, maybe you haven't noticed that before. Virion means the infectious virus particle. That is, if you have a 10,000 to 1 ratio of non-infectious to infectious, that one particle out of the 10,000 is the virion. The others are all particles. Yeah, a virus which may or may not be infectious. So the particle-to-PFU. Particle is everything, both infectious and non-infectious viruses. And the PFU are the ones that are infectious. So that's why particle can be both. And a bunch of you got infectious, but the particle and the particle-to-PFU, that particle, if it's 10,000 to 1, not all of those are infectious. That's why you can't say it's infectious or not infectious. It's either one, and that's encompassed in B. All right, let's talk about studying the virus infectious cycle, which is the name of this lecture. How do we do that? And this is called the one-step growth cycle, which was first developed in 1939, again, by people working on bacteriophages. And what you do here is you take your virus preparation, you adsorb it. That's a word we use when we say we add virus to cells and let the virus stick to the cell. We then dilute the culture so no more infections occur. We synchronize the culture, and we then take samples at different time after infection, and we measure virus, say, by Plaque assay. That's a one-step growth cycle. We're going to take a culture of cells, whether they be bacterial or eukaryotic. We're going to infect all of them. We're going to let the virus replicate and end, and then we're going to see what that looks like in terms of production. And we actually looked at a version of this last time. So here on the left, we have a one-step growth curve where we have infected cells here, and we dilute the culture so no more infections occur, so it's synchronized. And then we're looking at infectious viruses produced over time. And remember, there's an eclipse period. The eclipse period, nothing seems to be happening in terms of the number of infectious particles produced. But in the cell, the genome has come out of the virus particle, and mRNAs are being produced, proteins are being produced, and eventually virus particles will be assembled. And when those first particles are made, then we see viruses coming out of the cells. And that's when the curve goes up until it plateaus when all the cells are dead. So this we call the burst or the yield. And the two key parts here are the eclipse, where apparently no infectivity is being generated, but in the cell lots of synthesis is going on. And then when the first virus particles leave, we have the burst. So this is a one-step because all the cells are infected. They go through the same phase of the replicative cycle, and they all release viruses at the same time. So in terms of studying these cells, you can imagine that's good. Every cell is giving you a signal, and it's amplified so you can figure out what's going on. Sometimes you can get multi-step growth cycles, and there are reasons why you might want to do that. Here, you infect fewer cells to start with. Let's say you infect 10% of the culture. Those viruses will release, those infected cells will release virus. That's the first burst. That virus will then go out and infect neighboring cells, and then you'll have a second burst as well. So depending on how much virus you initially put in the culture, you'll either get a one-step growth where all the cells are infected, or you'll get multi-cycle where only a fraction of the cells are infected at any time. So here is now a one-step growth cycle for an animal virus. This is Adenovirus, a large DNA virus that we'll talk about somewhat. And again, you infect cells, you dilute the culture to prevent more infections, so you have an eclipse period. And then here, about 12 hours, you start to see the release of infectious viruses. Here, we're looking actually at intracellular virus particle production. So we're breaking open the cells at each point in time, and we're measuring the infectious virus. But still, we can define an eclipse period where there's no infectious virus followed by the burst. Now, if you take this one step further, if you look at virus in the medium, that's extracellular virus, you can see there's a lag between the time when the first infectious viruses can be detected in the infected cells and when they're detected in the medium. Okay, and that comprises the latent period between time 0 and, say, here, time 16. So the eclipse is when you can't find any infectivity in infected cells, and the latent is when you can't find any infectivity outside of the cell in the culture medium. So this tells you that there's a lag between making an infectious virus in a cell and it getting out into the medium. And I want to contrast it again with bacteria. We did this last time. With bacteria, you put a single bacterium in broth, it begins to divide almost immediately, and you have logarithmic growth. And that's because bacteria divide by binary fission. Viruses don't do that. They get in the cell, they have to express their genetic information, make the parts to build new virus particles. That takes time, and that's the eclipse period. That's that time when you can't detect any infectivity. Go back here, eclipse period. And its length varies depending on the virus. You can see it's pretty long here, 12 hours. There's some viruses, polio, it's 2 to 4 hours is the eclipse period. It doesn't take a long time to assemble those first infectious viruses. So that one-step growth cycle, we have all the cells infected, they all go through the cycle together, they're synchronized. How do we know that we're infecting all the cells? Is it just hit or miss? Well, of course not. We have to know how much virus we're putting on. So you have to know how much virus you're putting on the cells because you want to get every cell infected. And the key to understanding that is called multiplicity of infection. That is very easy. The number of infectious particles you add per cell. Infectious particles, that is multiplicity of infection. It's not the same as what each cell receives. It's the number you add. So for example, if we add 10 to the 7th infectious virus particles to 1 million cells, the MOI is 10. But that's not what each cell receives. Each cell does not receive 10 virus particles. What each cell actually receives is a distribution, which is based on the Poisson distribution. Think of it this way: if I had 100 tennis balls and I threw them all out at you, you wouldn't each catch one. Some of you would catch none. Some of you would catch one, some 2 or 3. Same idea. When you throw viruses on cells, if you throw 10 viruses per cell by your calculation, not every cell is going to get 10 viruses because it's a random event, how each virus hits a cell. And that's because infection depends on random collisions between viruses and cells. If I throw tennis balls at you, the chance that you're going to pick one up depends on random collisions of the balls with you. So when you mix virus with cells, and they have to be susceptible, of course. They have to have receptors so that they can attach to the virus. Some cells will still be uninfected. They don't get a virus. Some get 1 virus, some get 2, some get 3, some get more. And that distribution is defined by the Poisson distribution for virus infection of cells. And so that's how you know what fraction of your culture is infected, by using this distribution. And here's the formula. It's very simple. It's got pK on one side. pK is the fraction of cells infected with K virus particles. So K is the number of virus particles, and there's several Ks here in this formula. M is the multiplicity of infection, and E, of course, is the natural logarithm. So an example of this, we could set K to 0. If you want to know the number of uninfected cells in a culture, you set K to 0, and the equation works out to e to the minus m. And if you want to know how many cells get 1 particle, P1 is me to the minus m. You could put these numbers in and work through, but you don't need to do that. And cells multiply infected would be represented by this formula. And that is obtained by subtracting from 1. 1 would be the sum of all probabilities for any value of K. The probability is P0 and P1. That's how you would get P greater than 1. So let me show you a couple of examples. If you have 1 million cells and you infect them at a multiplicity of infection of 10, you could plug that into the formula. 45 cells are uninfected. So that's not a lot. Most of the cells are infected. 450 cells get 1 particle, and the rest get more than 1 particle. So most of the cells, the vast majority of cells are infected. So if you want to do a one-step growth curve, you use a multiplicity of infection of 10. 5 will also work. And if you know the titer of your virus, that's really easy to figure that out. That way you know every cell in the culture is going to be infected. If you use an MOI of 1, 37% of the cells are uninfected, 37% receive 1 particle, and 26% receive more than 1. So sometimes you'd like to have more than 1 cycle of virus multiplication. You would do an MOI of 1, or you could do an MOI of 0.001. If you want many cycles of infection, you can see 99.9% initially are uninfected. And then, but a few are infected. Some get 990 cells get 1 particle, and very few get more than 1. So a few will be infected, they'll go on to release virus, and you can see multiple cycles. So that's how you work out the conditions of the growth curve. You can either have a one-step at a high MOI or multi-step by using low MOI. All right, our question is, if cells are infected at an MOI of 10, In a one-step growth experiment, the growth curve, you will likely see multiple bursts of virus infection, multiple eclipse periods, a single burst of virus release, no burst of virus release, asynchronous infection. MOI of 10. All right, what do we got? 91% of you got a single burst of virus release. That's right? It's a high multiplicity of infection. Most of the cells are infected, so you're going to get one synchronous infection. Some of you said multiple bursts. That wouldn't happen at a high MOI. That would happen at a low MOI when not all of the cells are infected. And the others, no burst. Well, if you infect the cells, you have to have some kind of burst and asynchronous infection. The whole key to a high MOI is to get synchronous infection, which means that all the cells are infected at the same time and they all go through the replication cycle. Just think of all those million cells. Each one is going through the cycle pretty much the same as a neighbor. So we've talked a lot about measuring infectivity, particle-to-PFU ratio, MOI. Let's talk a little bit about measuring not infectious particles, because there are a lot of assays that do this. And here we use a variety of different assays, including hemagglutination. We'll talk about electron microscopy. You can simply count the particles. Sometimes we measure viral enzymes. Sometimes we use antibodies to measure viral proteins, and we can measure nucleic acids. All of these are ways of measuring particles, not telling you about infectivity, though. Hemagglutination is a very old assay. It's based on the fact that some viruses, including influenza virus, will bind to a sugar, sialic acid. That's their receptor that they use to get into cells, that happens to be on the surface of red blood cells. And so if you take red blood cells, chicken or guinea pig, human, you can wash them and get nice clean preparations. You add virus, so here's a little influenza virus particle. The influenza viruses will stick to the red blood cell, and then other red blood cells will stick to them, and eventually you'll get a nice lattice form, depending on how many virus particles are present. You can imagine if there are not a lot of viruses, you're not going to get red blood cells sticking together. If there are a lot, you will. So here on the top is an example of this assay, and these are individual wells where we've put dilutions of virus. So the 1 to 4 to the 1 to 8,000 is the dilution of virus. And then we've mixed those dilutions with the same amount of red blood cells. So what happens is after 30 minutes or so, the red blood cells will tumble to the bottom of a well and make a nice little button. So if there's no virus present, you get this button. So the top is clearly a sample with no virus in it. You get buttons in every well. There's no virus to prevent the button formation. If there is virus and you make a lattice, the lattice coats the side of the well. So you can see here, you're getting lattice formation. There's no button. And then at 1 to 1024, you start to see a button. So you've now diluted beyond the point where you make a nice lattice. So that's your endpoint for this assay. You can say the HA titer of this virus is 1024. It's the reciprocal of the dilution. So it's a very quick way. You can do this in 40 minutes or so of measuring the numbers of particles. It doesn't tell you anything about infectivity, but if you want a quick assay before you do your cell culture assay, this is a good one. You can also measure enzymes. Many viruses have enzymes in the particle, and one of them where this is very common is the retroviruses. These are viruses that have an RNA genome, shown in green, and within the particle are a variety of enzymes. One of them, which I'll show you as an example of measuring, is the reverse transcriptase. This is an enzyme we'll talk about at great length, which takes the RNA genome of the virus and makes a DNA copy. We can measure this enzyme activity quite readily and get an idea for how many virus particles are present. So here's an example of an assay for reverse transcriptase. We take, let's say, a solution with our virus in it. We add some radioactive substrate that the virus needs. We add a little detergent to permeabilize the virus, and then the substrate will get into the particle. The enzyme will copy it. We provide a primer and a template, and we can measure the amount of radioactivity incorporated. So here you can see these dots where the material has been filtered through a filter paper and exposed to X-ray film. These dots are a positive reaction for the enzyme of this particular virus. So you can make dilutions and you can make this quantifiable. And this is done, nowadays it's done without radioactivity as well, but the principle is the same. We can also measure antigens using antibodies. This is very commonly done. Enzyme-linked immunosorbent type assays. You can look for either viral proteins or antigens, or we can look for antibodies to see if someone has been infected with a virus. We could look for antibodies in your serum. And here is how these two assays would work. On the left, we're looking for a viral antigen. So we're taking some serum from you. We want to know if you're infected with a particular virus. You start with a plastic plate to which you've attached what we call a captured antibody. And you then incubate that with your sample. And if there's viral protein present, the antigen will bind to your first antibody. And then you add a second antibody to your viral protein, which has an indicator that you can readily measure, an enzyme or a light-emitting source. And if there are antigens in your sample, you'll get a positive signal here. So it's kind of a sandwich assay, if you will. You're putting your first antibody on the plastic, then you're flooding it with your clinical sample, and then you put a second antibody on. You can do the same idea with looking for antibodies. In this case, we put viral antigen on the solid support. We flood this solid support with your serum, for example, which has antibody in it. If it's got antibody to this antigen, it will bind, and then we can detect that antibody with a second antibody linked to an indicator. So these are often used both in research and in clinical applications, trying to find out if you're infected with a particular virus. Many of these have been developed into rapid assays, which can be done in physician offices, for example. Here's an example of what we call a lateral flow assay, which uses the same principles as I've just told you of enzyme-linked immunosorbent assay, but you do it in a format that can be read out readily. If you've ever seen any of these home pregnancy tests where you put urine on one side of the apparatus, and it flows through and gives you a color on the other end, it's the same idea. Here we're doing the same thing. We're looking in a clinical sample. We're asking if there are viral antigens. Are you infected with a particular virus? You would put the clinical sample on the sample pad at one end. It then wicks across by capillary flow on this filter. And what's embedded in this device are antibodies to your viral antigen. They're linked to colloidal gold, so you can see them readily. They move down, and then here we have a row of antibodies attached to the device, which are to the antigen. And eventually, if you have antigen in your serum, that antigen will bind those antibodies, shown in the bottom, and then your primary antibody will also bind the antigen. You'll make a sandwich. And you can see this quite readily by the colloidal gold. You can actually just see it visibly. You could also do a color assay if you wanted to. And then there's always a control line to show you that the test is valid. In this case, we're pulling down, we have antibodies to the actual antibody itself that is being used in the test, just to show that the test works. So there are lots of variations of this that are now in use, which make it very easy to do all sorts of diagnostics, not just in offices, but in the field. You can go into the backcountry and do these these assays as well. We use fluorescent proteins a lot in virology. Of course, green fluorescent protein came from a jellyfish originally, and it was shown to be a great way to track proteins in cells of various sorts. So there's the original jellyfish from which it was derived. And many, many color derivatives have been since made by chemically altering the original GFP. And here's an example of a virus infection. These are cells infected with, I think, 7 different herpes viruses that each encode a different color fluorescent protein. And you get way more than 7 colors here because you get different combinations of viruses infecting different cells. And so that was a revelation that we could understand how many viruses were infecting a particular cell. So each of these has a unique kind of color made by combinations of the different GFPs. One of the really neat applications for these fluorescent proteins is that now we can do single virus particle tracking. We can see, because we have great high resolution microscopy and we have these fluorescent proteins which basically amplify the size of a virus particle, we can see single virus particles tracking. So here on the left, GFP has been inserted into the genome. The gene encoding GFP has been inserted into the genome of a herpes virus. And this virus was used to infect the brain of animals. And you can see that this neuron is full of GFP because it's virus infected. And so you could actually inject this virus at a very particular place in the brain and trace all the neuronal connections by looking at where the GFP is going throughout the brain. That's an example of that. On the right is a cell infected with HIV particles. Those are the green particles. And these have been labeled so they incorporate green fluorescent protein into the virus particle. And this is actually the microtubule network of the cell, these red filaments there. And you can watch individual virus particles. You can do live imaging and watch individual virus particles moving along these microtubules as they come into the cell. So this is incredibly powerful. We can see individual viral particles in infected cells. PCR has been another boon in many areas, of course. Research, industry, diagnosis, and polymerase chain reaction, of course, depends on the use of a thermal stable DNA polymerase, which came from originally from a bacterium that lives in the hot springs in places like Yellowstone, where the hot grasses that we talked about also live. And a microbiologist in the 60s named Thomas Brock, he was just interested in these bacteria that grew there. You can see the different colors are due to them. And he isolated a whole bunch of them, one of them Thermus aquaticus. And many years later, someone purified the DNA polymerase from this organism. Of course, it turned out to be thermostable. You could get polymerization at incredibly high temperatures. And that enzyme and many others that followed turned out to be the basis for polymerase chain reaction, where you can take a very small amount of nucleic acid and amplify it and be able to detect it. And that's shown here. You have a piece of double-stranded DNA. The way the reaction works is you denature it. So you have two strands. You anneal the strands to a primer, a set of primers. You then extend the DNA on the primer with your thermostable polymerase. You then denature those, and you go through multiple cycles of denaturing, polymerization, denaturing, polymerization. That's why the enzyme has to be thermostable, of course. And you go from one molecule to 2 to 4 to 8. You get exponential amplification with each cycle. So after 40 cycles, you may not be able to detect the original DNA, but you can certainly see it now on a gel. And this has been great for detection, clinical uses, research, producing genes, and so forth. So every lab now does this. It's a mainstay of virology. It has totally revolutionized, of course, not just virology, but every field as well. But I just want to give you a caveat with PCR that a lot of people don't think about. And that is that a PCR product is not the same as infectious virus. All you're looking for is a short piece of DNA made with your polymerase on a template. It's a piece of a genome. We never amplify the entire viral genome. It's very inefficient to do that. Of course, some virus genomes are huge. So we're only looking at a little piece. So when you have a diagnostic done for a virus infection, you have some symptoms, you go to a physician, they do a PCR test for influenza, it comes back positive. You're probably infected because you have the right symptoms, it's the right time of year, and so forth. But in research, you cannot conclude that a PCR positive result means there is infectious virus present. A lot of people get misled over this. So let me give you an example of how this works. This was an experiment done with Zika virus in mice last year, 2017. And the title was Frequent Zika Virus Sexual Transmission and Prolonged Viral RNA Shedding in an Immunodeficient Mouse Model. What they did here, they wanted to understand sexual transmission of Zika virus in mice. They infected male mice subcutaneously, and then at different days after infection, they took seminal fluids and they looked for infectious Zika virus. How would you look for infectious Zika virus, you think? What's an assay you could use? What's an assay for infectious virus? Plaque assay. My favorite assay. You can't go wrong by saying plaque assay. So you can plaque Zika readily. It makes nice plaques. And look, you get a peak of infectious Zika, I don't know, about 15 days post-infection, and then it is gone. No more infectious virus. But guess what? If you look at PCR of the seminal fluid, the Zika virus is an RNA virus. You can convert the RNA to DNA by reverse transcriptase and then do a PCR. So you do that. Look, 60 days, you're still detecting Zika RNA. And many people, when this was done in people, they took it to mean that you're infectious for that many days with virus after you were infected with an infected mosquito. But that's actually not true. At least in mice, it's not. I haven't seen the same study done in people. But this clearly shows that just because you have a PCR positive test does not mean that there is infectious virus present. And people make this mistake over and over. So just be careful when you're looking at a research project, which is what this is. We're trying to figure out how long the virus persists in seminal fluid. You have to look at both infectious virus and PCR. But most people don't want to do a plaque assay because it takes a lot of work, whereas PCR is really easy. Now, the bottom experiment is interesting. If you vasectomize the mice, the infectious virus is reduced and it also goes away earlier. So obviously, vasectomy can prevent transmission. However, I don't think that's a good means of preventing Zika virus spread. There are other ways that you can do it more readily. Okay, PCR product is not the same as infectious virus. Let me leave you with one more story. We now can do incredible, deep, high-throughput sequencing. And that means you can, let me back up. When I was a student in postdoc, we did sequencing like is shown here. You would do reactions on nucleic acids and run them out on gels, and you would read the gels by hand. And you were lucky if you could do a few hundred bases at once, and your coverage was very low. That is, you got one read per nucleic acid molecule. Now, different methods have been developed, which are just brilliant in the way they work. We don't have time to go into them. But they allow you to get hundreds of times coverage on a single molecule, and you can get many, many different sequences. So you can take a sample of anything and just sequence all the nucleic acid in it and get lots of information. That we call metagenomics. You can do things like identify new viruses in the environment. You can identify new pathogens. This has made sequencing the human genome cheap. The first human genome sequence took 10 years. It cost $3 billion. You could have your sequence done tomorrow for $1,000 because the technology has increased so much. And we're going to talk a lot about the uses of this technology in virology. You can basically know all of the nucleic acid sequences that are present in a sample with reasonable certainty. These methods are amazing. Two examples of recent applications. These are two episodes of This Week in Virology. Here on the left, they discovered a new virus, a pathogen of snakes. So this was done in Joe DeRisi's lab at UCSF. And on this episode, he was there and he told this story. A woman sent him a picture of her snake who had died. And she wanted to know what was wrong with it. And he said he forgot about it. She sent him a picture of the snake around her neck when it was living. He forgot about it, but then for some reason he was talking to a veterinarian and this came up. So they got snakes with similar diseases and they extracted nucleic acids from them. They sequenced it. They found a new virus that's causing this disease in snakes. And you don't have to grow it in culture. You can just sequence and see, in most of the samples this sequence is present. So that's that one. It was an arenavirus. And then here on the right, there have been recently outbreaks of tick-borne disease in farmers in the Midwest of the US. So a few farmers got this high febrile disease with other symptoms. They all had many, many ticks because they worked in the field. And so they did some sequencing on the blood and they isolated what they call Heartland virus, which was, again, by deep sequencing the blood. This is what you can do now to identify new pathogens. So pathogen-driven virus research is driven by high-throughput sequencing. And finally, to get back to the PCR issue, and all this involves sequencing, of course. If you make a small PCR fragment, you usually sequence it and say this virus is present or not. This is a paper published a number of years ago, zoonotic viruses associated with illegally imported wildlife. So lots of people try and get all these dried out animals and animal parts into the country. I don't know why, but they want to keep them as relics. Of course, they're confiscated at JFK when they come in, or Newark Airport. And these are some of them. their arms and legs and a head there. And my colleague at Columbia, Ian Lipkin, got some of these and he extracted nucleic acid and he did some sequencing. And so he found some partial sequences of viruses. And if you look at the paper, you can see the list of viruses that are associated with these products. But my contention here is that they're not viruses. So the original PLOS One article said zoonotic viruses, and the New York Times covered this story. Viruses cross borders in monkey meat. I would argue there's no infectious virus there. They didn't look. They just found pieces of DNA, and that's not the same. And you can argue that maybe there is some virus there, but until you show it, you really can't conclude that there's infectious virus present.